The goal in our project was to combine magnetic resonance guided focus ultrasound and radiation therapy for the cancer treatment in a mouse model. I want to present you the developed and used focus ultrasound system. Here you see the whole system for MRI-guided FUS in small animals. The probe consists of 11 by 11 elements matrix array working at a frequency with around 1 MHz or another transducer with 2 MHz. For MRI-Compatibility, the probe has an inner copper shielding. On the screenshot, you see the software for the treatment planning by using the sound field inside of a preclinical MRI animal bed. For testing the transducer in the preclinical MRI, the transducer was fixed with tape on the red animal bed of a 7 Tesla Bruker MRI, together with an imaging phantom. After adding the coil, the whole setup was inserted into the bore. MRI compatibility measurements included field mapping of the static magnetic field B0 and acquisitions with multiple pulse sequences, RAR and flash. The signal-to-noise ratio was influenced by the FUS transducer. Overall, there is a 92% signal loss when the transducer was placed on the animal bed inside the MRI. For real-time temperature control during a FUS treatment, we established the proton frequency shift thermometry in the Bruker. To start this, we used a tissue mimicking phantom and measured the temperature with fiber optics in parallel to the PRF with the MRI. Our PRF thermometry measurements conforms with temperatures of the fiber optics during the sonication in the phantom. After using the tissue phantom, we established the PRF in vivo. Figure A shows the position of the transducer, the agar pad for coupling, and the tumor in the magnitude image. The xenograph tumor had a diameter of 1.5 cm along the major axis. A heating pad was placed below the animal to keep the body temperature stable. Using the 2 MHz transducer, the sonication power was kept at maximum for the first 2 minutes, so that the temperature of the targeted tumor was raised from 37 to 45 degrees. Afterwards, the power was reduced and varied between 10 to 15%, so that the temperature was stable at 45 degrees for 30 minutes. Since our goal is the combination treatment in vivo in tumor-bearing mice, we established the xenografts by subcutaneous injection of the human prostate cancer cell line PC3. For the experiment, the mouse was anesthetized with isoflurane and placed on a warming pad. The MRI conditional transducer was positioned on top of the subcutaneous tumor, and a phantom pad and ultrasound gel was used for coupling. And then the FUS heating was performed at 45 degrees for 30 minutes. Following the FUS treatment, a single dose of X-ray was added to the tumors. Here you can see the results for the tumor volume in the different treatment groups and the survival probability of the animals. And you see that in vivo, the tumor growth of the prostate cancer cell line PC3 was inhibited after the combination of focused ultrasound and radiation. Furthermore, we analyzed the proliferation and also stained the cell nuclei of the tumor after treatment. And the combination of FUS and radiation leads to membrane damage of the tumor cells and to a reduction of the total tumor nuclei number. Finally, we tested the proliferation and the impact of the combination treatment on apoptosis. And we saw that the proliferation was reduced in the combination group compared to apoptosis, which was increased. For the future, we're working on a second generation for the small animal therapy system with new electronics and other software modules. Thank you for listening.